Hello, everyone. I'm Dorsa, and today I want to present to you my master's project. It's going to be a short presentation. The title of my presentation is Longitudinal Monitoring of Physiological, Psychological, and Molecular Responses to Diets, a Precision Health Approach. Just as an introduction, we all know our lifestyle choices, including diet, physical activity, sleep, and stress levels, are significant determinants of health. According to WHO, about 71% of deaths globally are attributed to non-communicable diseases, but about 80% of these diseases and their associated deaths can be prevented through healthier lifestyle choices. Such as avoiding tobacco use, moderate alcohol consumption, adopting a healthy diet, being physically active, and maintaining a healthy weight can significantly reduce the risk of developing these diseases. Generalized dietary and lifestyle recommendations are delivered as a common practice to motivate people towards making healthier choices. But differences in biochemistry, metabolism, genetics, microbiota, and lifestyle habits contribute to a significant difference between individuals in response to nutrition, and that's called intra-individual variability. Also, the response of an individual to a nutrition intervention can change over time based on external factors such as stress levels, sleep, and even aging. And that's called inter-individual variability. This suggests that one diet that might be beneficial for one person might not have the same impact on another. Therefore, to identify the optimal dietary factors for everyone, it is essential to move beyond the generalized one-size-fits-all dietary recommendations and adopt a more personalized approach. Precision nutrition specifically tailors dietary interventions to accommodate both inter and intra-individual variabilities, optimizing health outcomes. As an example, N-of-1 studies or single subject studies have been receiving interest within the past few years with the hope to shift towards a personalized health and nutrition approach instead of a generalized approach. But there are limitations to these personalized approaches. High costs being one of them. N-of-1 studies require expensive multi-omics platforms such as DNA sequencers, mass spectrometers, and extensive time investments. Studies often involve frequent blood sample collection and constant monitoring by healthcare professionals, such as doctors, nurses, and also it put a lot of pressure on the participants that they have to monitor themselves and go to clinics regularly. The absence of quantitative omics data in many of the current N-of-1 studies limits the ability to compare findings across different interventions. Overall, the current N-of-1 studies have not examined the effects of dietary or lifestyle interventions on mental performance and mental health. So therefore, the current model is not easily scalable due to high costs and research demands. With that in mind, the main objective of my project was to see if integrating wearable devices, quantitative omics techniques, and mental and physical performance testing would provide sufficient information to identify specific foods, diets, or habits that optimize individual health. I also had some secondary objectives, and I wanted to see whether quantitative omics data enhance the analysis and interpretation of N-of-1 nutrition studies, and if it's possible to conduct self-monitoring and biosample self-collection, and if it's possible to do it cost-effectively, and can monitoring mental and physical performance offer valuable insights in N-of-1 precision nutrition studies. So for that, I tried out 4 different diets myself, including a fast food diet, Mediterranean diet, ketogenic diet, and a regular diet. Each of the diets were for 2 weeks with a week of washout period between them to make sure there aren't any carryover effects from the previous diet to the next. All the diets were isocaloric, and I kept records of all the foods that I was eating, the amount of foods that I was eating in food diaries and food frequency questionnaires. The washout periods were my regular diet, but no monitoring was happening during those washout periods. During the fast food diet, I sourced all my meals from a chain fast food restaurant, and the main component of this diet was beef and red meat. So I avoided any kind of poultry or fish from those fast food. I included fries, milkshakes, sugary beverages such as Coke, and overall I had a high intake of processed foods, sugars, and fats. During the Mediterranean diet, I had a higher intake of fruits and vegetables, nuts and legumes, and a moderate protein intake that was mainly sourced from poultry and seafood, and lots of olive oil. During the ketogenic diet, I had a higher intake of protein and fat, and I limited my carb intake to less than 20 grams per day. And I had overall a higher intake of fatty cuts of meat, such as pork, and low carbohydrate vegetables. And my regular diet usually consists of a moderate consumption of carbohydrates and proteins, fruits and vegetables, and an overall lower consumption of red meat. And while I was on the diets, I used some monitors to monitor my body, including a body temperature monitor that continuously monitors core and skin temperature. I did gut microbiome testing after finishing each dietary intervention. I used a smartwatch to collect data on my heart rate, activity level, sleep. I also used a continuous glucose monitor and a blood pressure monitor. And I used the blood pressure monitor 5 times per day after waking up and after finishing each meal and before going to bed at night. I also used an at-home blood collection device called Paso Plus, and I collected fasting plasma samples daily, along with urine samples that I collected twice per day in morning and at night. Both plasma and urine were analyzed for metabolomics through using the TMIC mega assay, and then the fasting plasma was analyzed for pro-inflammatory cytokines through mesoscale discovery V-plex panel. I also did other kinds of testing, including daily measurement of my body weight, percent body fat estimation, and BMI. I monitored my physical activity, which consisted of jogging on a treadmill with a consistent speed and time across all diets for about 10 days per intervention. I also monitored my mental performance with reaction time, serial subtraction, memory testing, trail making, these kind of tests. And I also kept record of my mood changes through profile of mood states questionnaires. And I did genomics testing using health and ancestry kit from 23andMe. So overall, I monitored my body in 3 different levels using tests and questionnaires, wearables, and omics measurements. And the idea was to see how different interventions impact my body on these different levels and if there are connections and associations between these levels. And especially metabolome, if we can use that as a tool to cross-validate the data that we're seeing from different monitors or tests and questionnaires. So I have lots of data from each of these monitors, but because it will take forever to talk about them, I'm going to list some of the main findings and go into more detail for just a few of them. So overall, when I was on the fast food diet, I had irregular blood pressure patterns, decreased quality of sleep and mental performance. While I was on the ketogenic diet, I had weight loss, worsened mood quality, and gut health. Both on fast food diet and ketogenic diet, I could see increased inflammatory responses. While during the Mediterranean diet, I had improved cardiovascular health and mental performance. Some of the main findings in terms of analysis was that we could see most of the changes happen within a few days of the start of each intervention. The bigger part of the changes and the biggest shifts were happening within the first week of each intervention. I was able to see meaningful associations between omics measurements and mental performance and mood. I was able to cross-validate the data that we were seeing from wearable devices through quantitative omics, especially metabolomics. I identified many correlations of biomarkers of food intake, and I could identify food items that I have heightened glycemic susceptibility to. And I was able to validate sensitivity and specificity of wearables in detecting meal consumption and exercise episodes. Just as an example, I'm going to talk about the results that we could see from heart rate changes. We can see here that I had significantly higher levels of my heart rate, daily average heart rate, when I was on ketogenic diet and fast food diet, while it was significantly lower during the Mediterranean diet. If we look at the plot on the right, we can see lots of fluctuations during the regular diet in blue and fast food diet in pink, but we can see this increasing pattern during the ketogenic diet in orange, especially within the first week, and then it was more stabilized during the second week, but at a higher level. But again, during the Mediterranean diet here in green, we can see this decreasing pattern, especially during the first week, and then it was more stabilized during the second week, but at a significantly lower level. Another way to see the changes in heart rate and cardiovascular health is to see check the time it takes for the heart rate to drop to resting or near resting levels after finishing an exercise, and that's called heart rate recovery rate. The longer it takes for the heart rate to recover, the lower level of fitness or poorer cardiovascular health. If you look at the plots on the right, you can see that it took about 20 minutes while I was on my regular diet and ketogenic diet to come back to near resting levels, while it was about 10 minutes when I was doing on the Mediterranean diet. And even longer, about 60 minutes, an hour when I was doing the fast food diet. So overall, the rapid decline post-exercise heart rate while on the Mediterranean diet suggests an improved cardiovascular health, while the fast food diet suggests the reduced cardiovascular health, the slow decline that we are seeing during the fast food diet. Just an example of how we could check the connections between heart rate and metabolome. I grouped the days with very high heart rate together as high heart rate days, and the days that I had lower heart rate levels as low heart rate days, regardless of the diet type, and I compared those two together. And as you can see here, for example, on this heat map, we can see a shift in increasing in the abundance of fatty acids, especially ceramides, which are the lipids associated with lipid metabolism, and overall they promote inflammation, especially vascular inflammation. Ceramides can also cause endothelial dysfunction, and overall their elevated levels can increase sympathetic activity and put higher stress on heart, which leads to higher heart rate levels. Just to compare the levels through between the different diets, we can see higher levels during a fast food diet and ketogenic diet, and significantly lower levels during the Mediterranean diet. I also looked at the correlations between daily average heart rate levels and different concentrations of different metabolites, both in plasma and urine. And I have here example of the correlation that we could see with the concentration of ceramides and very long-chain fatty acid ceramides in plasma, and both of them showed significantly positive correlations. In another example, the data from blood pressure, the plots on the left, we can see the systolic blood pressure changes during the fast food diet, Mediterranean diet, ketogenic diet, and regular diet. And on the right, we can see the diastolic blood pressure during those diets. On x-axis, on the plots, you can see the 5 time points that I measured my blood pressure in the morning, after breakfast, after lunch, after dinner, and at night, right before going to bed. Overall, we can see a decreasing trend over the day for diastolic blood pressures for all the diets, but the systolic blood pressure was different. It shows different patterns between different diets. Overall, diurnal blood pressure trends normally exhibit a 24-hour fluctuation, with blood pressure rising in early morning, peaking mid-morning, then stabilizing at a higher level during the day, and then maybe declining slightly in the afternoon, and potentially rising again in the afternoon, in early evening, before bedtime. And during the sleep, blood pressure usually drops, known as the nocturnal dip, and that reflects the reduced sympathetic activity. And we could see that typical diurnal cycle during the Mediterranean diet and regular diet, but it was kind of absent during the ketogenic diet and fast food diet, especially during the fast food diet, that we can see this reverse dipping, that it's increasing and it's showing lowest levels during midday time. And all of this suggests the fundamental change in cardiovascular function during the fast food diet and ketogenic diet. Again, examples of the connections we could see to metabolome was the significant correlation that we could see between ratio of ornithine to citrulline in plasma and daily average systolic blood pressure levels. In the ureal cycle, ornithine combines with carbonyl phosphate to form citrulline, which is then converted to arginine. Arginine serves as the precursor to nitric oxide production, which is the molecule that is essential for vasodilation and vascular health. So therefore, a higher ornithine to citrulline ratio suggests an abundance of ornithine relative to citrulline and potentially indicating a bottleneck in arginine production, which this imbalance can lead to reduced nitric oxide synthesis and decreased vasodilation and overall elevating impact on systolic blood pressure, as we can also see here with the positive correlation. Another way that I looked at the data that I have collected was to see the biomarkers of food intake. Just a short description on biomarkers of food intakes is that some foods are rich or poor in specific compounds, and these compounds will increase or decrease in plasma or urine within a few hours after consumption. And overall, their concentrations have little to do with any kind of physiological states or pathways that are happening in the body. Oops. So as an example here, when I compared this plasma samples that were collected after banana consumption to the days that were following no banana consumption, we could see a difference in the concentration of endopropionic acid that was significantly higher after banana consumption. IPA is a plant hormone and is found in fruits such as bananas, but also it's a naturally produced metabolite that is synthesized by certain gut bacteria from tryptophan. There are other foods that are they have concentrations of tryptophan in them, including chicken, turkey, oats, milk, tuna, salmon. But when I compared the samples following consumption of those foods, we couldn't see this difference in the concentration of IPA or even tryptophan, suggesting that this might actually have something to do with the banana themselves. We could also see a correlation between the amount of banana that I was consuming with the concentration of serotonin in plasma, which was significantly positive. This could be linked to the pathway, the serotonin pathway, the tryptophan that is in banana can act as a precursor for serotonin, and it can be metabolized in the gut, and the presence of IPA actually shows an active tryptophan metabolism. I also compared the data that I collected from banana consumption, endopropionic acid, serotonin levels, and the data from mood questionnaires that I had, and we could actually see that negative correlation between serotonin and banana consumption and tryptophan and lower levels of depression or overall better quality of mood. Another example of biomarkers of food intake was the higher levels of trans-4-hydroxyproline that we could see in plasma after fast food diet. Overall, hydroxyproline is an essential component of collagen, a fundamental protein found in connective tissue, organs, and muscle in all mammals. Although I had the highest levels of protein intake and red meat intake during the ketogenic diet, still I had highest levels of hydroxyproline during the fast food diet. And this suggests that, in fact, the meat that I was consuming during the fast food diet was consisting of low quality ground organ meat with lots of connective tissues such as tendons, ligaments, and fascia. And it suggests that this metabolite could be used as a measure of quality of the meat, especially red meat. So just to conclude, I was able to identify the optimal diet and event-specific foods for myself by combining wearable sensor data, psychological and physical performance testing, and omics methods. The Mediterranean diet resulted in a better mood, mental performance, heart rate, and cardiovascular state. The fast food diet disrupted blood pressure regulation, possibly linked to increased inflammatory markers, and was characterized by poor sleep and mental performance. The ketogenic diet showed evidence of liver stress and high energy output, and high heart rate levels and oxidative stress. In terms of my secondary objective, I could see that self-monitoring and biosample collection in an N of 1 setting is feasible using a combination of wearable devices and lower cost omics measurements. In terms of cost effectiveness, while initial investments on the devices and sample kits and even the omics analysis are required, the temporal insight gain and the amount of data outweighs the costs. Monitoring mental and physical performance provided valuable data, with mental testing offering unique insights into cognitive responses to different diets, especially on Mediterranean diet and fast food diet for myself. And quantitative omics data, especially metabolomics, significantly enhance the interpretation of physiological changes by identifying biomarkers linked to specific diets. And for future directions, by aggregating results from multiple N-of-1 trials, we can develop more refined criteria for identification of subpopulations of individuals that share common genetic and phenotypic factors, which are called metabotypes, and this can be used to reduce the cost and increase the scalability of personalized approaches. And with that, I want to thank you for listening to me, and I'd be happy to take any questions.